Okay, well, thank you, Dr. Meekins. Yeah, and I did want a chance to expand on something that we are doing at Cal Poly. So I will have a few slides at the end, but for the most part, this is going to be well, a little bit in the weeds because that's kind of my personality. So first, let me tell you something about Cal Poly. Oh, see, there it is, that first slide. So what we are doing, or what I did in my postdoc, was try to disentangle this mess of things going on. And I will get to explaining what all is going on here. But if you want to follow along, the paper related to this was published in JACS in 2016. And then more recently at Cal Poly, working with undergraduates, we're looking at some different transition metal complexes, and specifically looking at how their light absorbing properties change when we somehow tune the functional groups on them. At Cal Poly, well, it's an interesting place. So for us, it's only undergraduate students. We actually have no graduate students here. So all of the research that happens at Cal Poly is actually from undergraduate students. If you come to San Luis Obispo, what you would see is this P, and this is called the Poly P. It's on the hill behind Cal Poly, and this is the Baker Science Building where my office and some of the labs are. If you climb to the top of this ridge and look out over San Luis Obispo, you can see into the valley here. And what's really neat is we're fairly close to the coast. I actually live way out there-ish. And so we're close to the ocean. We get some interesting weather, and it's generally pretty pleasant. In this picture here, it's a lot more green than it is right now. We did get some rain, but we're still kind of deep into that drought. But where we do chemistry is mostly in the Baker Science Building. So if you came to Cal Poly and came to the Baker Science Building, you could come visit. We have in here some innovative teaching areas. We have all of our general chemistry is taught in what's called a studio. So it's an integrated lecture and lab, and there are spaces that we've built that have clusters and computers at them so that we can do active learning and transitioning between different modes of instruction. And going up right now, and we're moving into it this summer, is a dedicated research building. So this is the William and Linda Frost Center for Research and Innovation. And it broke ground in 2019, and like I said, it's going to finish up soon. If you do come visit us, we're also going to direct you to some of the sites, and one of those sites is the State Park Montana de Oro, and I've had friends come visit. They're runners, and they really like going out there. You can run right along the Pacific Ocean and see some of the trails. You have to watch out for the poison oak, though, as my friend Eric would tell you. So getting into the science, what motivates what we do? Well, really the big question, and sort of an existential question, is, are we doomed? And that sounds kind of harsh, but we do have to think critically about, what's happening with the planet right now and how what we're doing affects it. There's an XKCD comic, so if you follow XKCD, most of it is stick pictures, but there's usually science interspersed. And one of the things that Randall Munroe proposed in this was looking at a temperature scale based on an ice age unit. Basically, what was the difference in temperature between today and the last ice age? Right now, we are headed toward one ice age positive from where we are, and I think it's actually a little bit further along the scale from when this was originally published. And one of the things we don't know is how much of a change we can expect in the planet as the temperature of the planet increases. The International Panel on Climate Change has published a series of reports, and this is the one from 2018. And in those reports, they do things like try to predict what sorts of changes we can expect on the planet as the temperature increases. When they apply those models, because they're trying to predict the future, they apply different parameters. So they may make assumptions under sort of a status quo. If we make no changes, what's going to happen? Well, we see this red line where we're projected by the end of the century to hit 4 degrees C, so roughly 8 to 10 degrees Fahrenheit higher on average temperature. And under that scenario, most of the planet is much hotter than what it would have been in the sort of average period that they're comparing it to. Even if we have some mitigation where we basically stop emitting within the next 20 years, we'd still expect to see significant change in temperature, but at least it would be mitigated and might be easier to adapt to. And so one of our objectives, as society in general, is to try and slow down the release of carbon dioxide to limit that. So how are we doing on this? Well, a current projection is we are going to cross what is assumed to be the threshold for where we can start to expect to see some changes. And there was this ideal goal of hitting 1.5 C and trying to keep it under the temperature of 1.5 degrees C. This was, like I said, published a few years ago, so the trajectory has gotten worse, and it seems the probability is that we are going to exceed 1.5 degrees C within the next 10 years. When I showed this figure to my students way back in 2018, so not that long ago, one of the things I pointed out to them is when I was sitting in the seats that they're sitting in, it was around 2001. So in the 20 years, we've seen sort of the trajectory just keeps going. And if we don't do anything, we really risk having some serious changes to our climate that we may not be able to adapt to. And based on where we are headed, we are tracking with some of the worst case scenarios right now. And so this is somewhat depressing because when you think about it, the whole world is basically a steady state equilibrium. We want to think about it as we have this incoming energy that comes from the sun and enough gets radiated out that our system is in balance. But by absorbing too much energy or it gets too warm, things heat up. And for anyone that knows the equation for an equilibrium constant, temperature is related exponentially to these equilibria. And so stuff gets shifted around. And sometimes that can have dramatic effects. So what can we do? Okay, after we get past the doom and gloom, what can we do? Well, the one thing that we can do, and what this talk is going to sort of allude to, is we can stop indiscriminately dumping carbon dioxide into the atmosphere. And we can do that by transitioning all of our power generation and consumption to renewable resources. For my interests, that renewable resource is the sun. Partly because in all of society's lifetime, there will be a sun and it will be irradiating the earth with light. And we can use that energy if we can capture it and funnel it. Now the question is, how do you capture it? Well, we could put devices on our roofs, and people do this, and maybe that's a strategy for doing it. We could capture that light energy and turn it into electricity. Or there are other things that we could do. We could capture the light and we could turn it into chemical energy. So we rely a lot on fuels, for example, for transportation. And the fuels, technically, those are already stored solar energy. They got stored, there was solar energy that was stored by plants millions of years ago, and over millennia and geological timescales, that plant matter got turned into oil, and then the oil got refined into gasoline and put it into cars and we could transport. What would be nice is if we could bypass that whole long time step and stop pumping carbon out of the ground and somehow use the light energy to sort of make those fuels a reality right now. So how we do that is actually related to an idea called photochemistry. Photochemistry in general, for anyone that isn't familiar with photochemistry, is we can perturb a system using light to get a result that's different than what we might get from a traditional reaction. So I have here just a schematic that shows this thing that's a reactant, and how we might do a chemical reaction traditionally is we put all our reagents into a flask, we heat it up, and we wait. And from that, we get some selection of products out of the flask, and we can get our whatever our chemical products are, and we can funnel those into whatever other thing that we want to do. Now, it's not always that we want to have our chemistry controlled by how fast things react, or what the thermodynamic products would be if we heat them for a long time. Instead, sometimes we might aim to have different products. And we can do that by shining light on the reactant, for example. If we can excite the reactant so that it has a new electronic configuration, then that excited reactant will sometimes have different photoproducts that result from that chemistry. And those different photoproducts, that different product profile, might be useful for driving a process. This isn't a new idea. In fact, there are a lot of different areas of research looking at using light to do things. So phototherapeutics, for example, in this top slide, this is from my PhD, and we would inoculate cells with this very large supramolecular complex, and anywhere where we shine light, we would see cells die, for example. And so this has implications for things like cancer therapy, where you can excite and kill cells specifically where you have light. There are other regions where we could do photochemical synthesis. So this is sort of a big area of research right now, photoredox catalysis, where you excite some dye, and then that dye can activate something toward a reaction that it's normally not reactive to. So in this case, this is from David Niskowitz's group at UNC. They can activate aromatic ether toward nucleophilic substitution. And there are other reactions too, where people have activated Diels-Alder, where you have non-compatible diene and dienophiles to do Diels-Alder cyclization, and other reactions as well. There are also times when you don't want photochemistry. And so if you have maybe textiles and polymers, right, we like having colorful clothes, but if you had your clothes on and you went outside and stepped into the sunlight and they disintegrated and fell off your body, that would be kind of a poor outcome. So being able to turn off an excited state so that you don't have photochemistry, that's also an interest in doing this. The thing I'm going to tell you about right now is using photochemistry to create solar cells using a combination of basically dirt and pigments. If you look at, so anyone that's in sort of photovoltaics or studying solar cells or anything like that, you've probably seen this figure. This is from NREL, and this is the device testing results that they have. And they have a lot of the different technologies shown here for solar cells. And you can see things like traditional silicon solar cells and what their efficiencies are. One of the problems with some of these, though, even though they have really high efficiency, is they're really expensive to make. And so what would be nice is if we had a way of processing things by maybe making a film that you can bake in the oven and then dipping it in a dye, and then that becomes itself a solar cell. And so down here, these open circles, some of the lowest efficiency at this time were the dye-sensitized solar cells. But what's really interesting about them is that they're relatively inexpensive. And some people may say, well, what about perovskites? And I'd say perovskites are amazing, and I can't wait to see what happens with them. And perovskites, for anyone that's followed that story, they've only been around for not even 10 years, I don't think, as a solar cell technology. And they've gone from 14% to upwards of close to 30% right now. So that's a really interesting technology. But anyway, I go off on a tangent there. So how does a photovoltaic work? So if you have a semiconductor, what makes it a semiconductor is there's a gap between the filled states, which we call the valence band, and the empty states, which we call the conduction band. The way that we can generate electricity using light is we can excite the electrons across this conduction band and those electrons get funneled through a circuit, and this voltage difference then can be used to drive whatever sort of device we have. Sorry, this was work I did with John Papanikolaou, or I was actually part of a team here, and Michelle Gabriel and Justin Kirschbraun actually led this. Eric Rumstrup's now at Montana State. He's a professor there. And the experiment, what involved this team of researchers was James Cahoon's group. So that was Chris Pinion and why am I blanking on his name? Jay Christensen. They generated these silicon nanowires, and then we put them under a microscope and we would excite them, and you could excite just a region with an ultrafast laser. It generates a population of electrons and holes, and then in these nanowires we actually had regions of n-doped and p-doped, so that it created an electric field and would separate those charges into opposite regions of the wire. And so you can see, as we increase time, you can see that original population sort of spreading out, and then the electrons going one way and the holes going the other. But silicon as a material, it's great as far as photovoltaics go. It absorbs a big chunk of the solar spectrum. So this is the solar radiation spectrum, and silicon absorbs a big chunk of that, so it can convert that into usable electricity. One of the problems is, to get silicon that can do this takes a lot of energy. You have to basically turn sand into something like like a thermodynamic sink. Take sand, remove all the oxygen, and then recrystallize it into a single silicon crystal. And so what would be nice is if we could take something that's essentially dirt, like I said, dirt, and use that to make our photovoltaics. Components of dirt are basically metal oxides. And so we have things like nickel oxide, tin oxide, titanium dioxide, zinc oxide, or zirconium oxide. And all of these have different properties that maybe make them more or less favorable for a photovoltaic. And with one of the ones that I'm going to look at is the titanium dioxide. But one of the challenges here is, if we look at this band gap, the band gap is sort of the minimum energy that it can absorb. So if you look at where titanium dioxide absorbs, it absorbs in the ultraviolet region, but it doesn't really absorb a whole lot of this solar spectrum. And so what would be nice is if you can somehow modify the TiO2 to be able to absorb more of the visible spectrum and then use that in a photovoltaic. So Brian O'Regan and Michael Gretzel did this in '91. This was sort of the seminal paper on dye-sensitized solar cells. And what they looked at was called the photoaction spectrum. So basically looking at the photo voltage based on the wavelength of light that they used to excite it. And pure titanium dioxide, like the solar spectrum shows, it absorbs in the UV region, but drops off really quickly. But if they attach a dye, in this case a ruthenium dye, to the surface of the TiO2, what they see is that action spectrum shift further to the red. So it's absorbing more of that ultraviolet light and turning it into electricity. The structure of the film itself is actually nanoporous. So it's tiny little crystals of TiO2, where the pores are very small, on the order of just tens of nanometers. And we've over time, people have gotten really good at making films of TiO2 nanoparticles. So this is one from Bertolucci's group. And you can see that there are a lot of pores there. This thing is roughly 14 microns thick, but each of the little pores there is gaps in this. And then there are channels where materials can diffuse in and out. But what we'd really like to do is be able to stick those TiO2, the dye, onto the surface of those. So how does it work? Well, with the dye sensitized solar cell, we have our dye. It's a molecular dye. It absorbs usually in the visible region of the spectrum or near IR. We aim to catch as much of the solar light as possible. We excite the dye, and the dye excited state can then inject an electron into this set of empty states on our conduction band. And if it's allowed, thermodynamically allowed, we'll get something called what we'll call electron injection. And there are a lot of words here, but this is basically just showing that we have a redox couple where we can consider the excited state and then the charge separated state after it injects electron versus the ground state and the charge separated state there. If we connect this thing into a circuit, we can close the loop by putting in a redox couple. So this is just something dissolved in solution that can be reduced. It can dump its electrons off to the oxidized ground state dye, regenerating our ground state dye, and then our dye can absorb again, and we can have all this stuff happen. For the purposes of this talk, I'm going to focus mostly on the electron injection side, but I figured some people may not have seen the photophysics and how we talk about excitation here. And so I like to give a primer on photochemistry every once in a while because I like photochemistry. So this is one I did recently and most people liked it, so we'll see how it goes. When we think about excited states and ground states, we talk about them in their relative energy. So on that last figure, I had just 2 lines at different energies, but we might consider some additional information there. And that is the processes that go into changing one state to another. Now, when I'm talking about a state, I'm talking about an electronic state. So an electronic ground state has an electron configuration that is the lowest energy electronic configuration. If we shine light of appropriate energy on our ground state, then we can change the electronic configuration, which changes the energy of the state. And the difference in energy between these 2 states is equal to the energy of the light that was absorbed. We may have more than one excited state, and so we might populate a different excited state with a different electronic configuration, and that'll correspond with a different energy of light as well. Alternatively, we don't have to always have the spins in the same state as the ground state. We can undergo processes where we generate different spin states, so our electrons have different spin states, and these would be considered a different set of excited states. Naming these states, I'm going to refer to them if all of our electrons are spin paired, so all of their spins are opposite each other so that we have no net spin. I'm going to call that a singlet state. And if we have any that are spin aligned, with 2 spin aligned, or net 2 spin aligned, I'm going to call that a triplet state. And all of these states can interconvert with each other through a variety of processes. If it interconverts with no change in spin, we call it internal conversion. If that happens with release of a photon, we call it fluorescence. If it undergoes a process where we have a spin flip, we call it intersystem crossing. And then we can have photochemistry or electron transfer, electron injection. Now, one of the things we can notice about our electron configuration here of the excited state is that it itself has a higher energy electron, which makes it a better reducer than the ground state, but it also has a lower energy vacancy, which makes it a better oxidizer. So one of the things that people like to say is that the excited state is both a better reducer and a better oxidizer than the ground state. And we're going to take advantage of this fact that the excited state dye is a better reducer and a better oxidizer to drive our photochemical reaction at the interface with the TiO2. So to be able to make these measurements, we have to first make our devices. And the way that we're going to do that is start with a conductive glass. So we have a film of fluoride doped tin oxide. We call it FTO. These are actually fairly inexpensive. And then we can put a paste of our TiO2 and we doctor blade it on. That was my stylized razor blade. But actually, when we do it, we use a Pasteur pipette and Scotch tape. Works really, really well. After we make the paste, we let it dry. So we let it air cure, and then we bake it to be that sintered film. Once we have that sintered film, we can immerse it in a solution of our dye, and we buffer the dye with an acid solution so that we can get formation of a chemical attachment of our dye to the surface of the TiO2. Then we rinse off the excess dye by immersing it in just a clean solution. And once we have this, we get a film that looks something like this. And these films are really stable. When I was at UNC, we regularly had films stored under solution conditions like this that were stable for months in the dark. Okay, so we have our device. Next, if we want to learn more about the electron injection process, we have to figure out how we're going to measure it. And the measurement we're going to use is something called transient absorption spectroscopy. And I think Dr. Meekins might like this part of the talk, especially because he said this is something that he did, and I dabbled in it a little bit at UNC. So what we can do is try to understand the electronic states of our dye by shining light on it. So we could just shine light of all colors on our dye, spread the light out, and measure it in a detector, and we get an electronic absorption spectrum. Some people call this a UV vis, but I like calling it an electronic absorption spectrum because it gives us information about the transitions between ground state and different excited states. Now, this is really useful for these direct excitations, but sometimes our photochemistry happens from states that we can't see be directly excited. And so one of the things that we can do is actually add a light source that precedes our probe light here, and we can generate a population of the excited states. So when we do that, we can send our pump light in, generates a population of excited states, and we get an absorption spectrum that looks different. That absorption spectrum has two things. One, since we have fewer of the ground state around after we pump the sample, we get some bleach here. So we can see a decrease in the absorbance that corresponds to bleach of the ground state. We also get changes, and these are a little harder to see in absorption, and those absorptions correspond to direct excitations of our excited state to some higher excited state as well. Now, this is an awfully subtle effect, and so what we choose to do is actually look at the difference spectrum. So we look at the difference relative to the original spectrum, and it looks something like this. And I have this normalized, so that's why the scales don't really match here. What we see is a bleach of our ground state, and we see an absorption that corresponds to the characteristics of the excited state here. Now, on TiO2, we actually see this band as we change the time between the arrival of our pump pulse and our probe pulse. We see a decrease in this band over time, and it turns out this is actually the signal that we can follow that tells us that we're having electron injection. So how would we think about this in terms of like populations? So imagine you have a nanocrystal of TiO2, and on the surface of it, we have a bunch of dyes. So these are all dye molecules. And we flash the sample, and immediately after we flash the sample, we generate a bunch of excited states on the surface. Now, one thing that we know from other experiments is that pretty immediately, within a picosecond, so that's 1 × 10⁻12 seconds, we have injection of a population of electrons. We still have some excited states on the surface, but we also have now built up this population of oxidized dyes that have injected their electrons into the TiO2 conduction band. Over longer time, we see more of the dyes have injected, and then after about a nanosecond, so this is 1,000 times longer than the first time point, but still a billionth of a second, we see pretty much total injection of all the electrons. And so we have a lot of oxidized dye on the surface and our injected electrons. And then if we wait long enough, it'll actually recombine and go back to our original state. One of the questions we have, though, is if you do an oxidative quenching experiment. So you take a solution of dye, you excite it, and you put in some quencher that can accept an electron from it. Those kinetics are usually single exponential. So why on the surface of this nanocrystal are they not? Well, we would have to somehow change the dynamics at the interface here. We somehow have to change either the relative energetics of the excited state relative to the conduction band, change the relative density of states in the conduction band. There are a number of parameters that we can adjust, and we decided to do an experiment where we tuned the energies of these excited states. So using that original figure I had with the conduction band of the TiO2, imagine if we took the dye and we made the dye a better reducing agent in the excited state, or an even better reducing agent. Like, how does that affect the injection process? The dyes that we chose, and this wasn't entirely a new idea, but one of the things that we wanted to focus on was making sure that when we change the parameters that affect this injection rate, we don't change anything else. We want to make sure that how the dye attaches to the surface is always the same, so that we don't perturb what's called the coupling between the excited state and the charge separated state. And we also didn't want to affect the reorganization energy. So when you undergo charge separation, you generate a dipole and so solvent and ligands, how those rearrange around the metal. There's an energy associated with that. And we'd like that to be consistent between our systems too. The way that we found to do that, and this is one of the neat things about working at a DOE research center, is we generated a whole bunch of very similar dyes. And so these are all ruthenium-based dyes, and this was at UNC, so it's kind of no surprise that it's ruthenium-based dyes. But what's really neat about these dyes is we can substitute just some of the ligands. We can leave anchoring groups on here, but change some of the ligands, which then tunes the relative energy of those excited states. So I have a lot going on in this slide, but I have 12 dyes, and it's actually 2 sets of 6. And the only thing that we varied for each set of 6 was what the substituent group was on this ligand. So it could be electron donating or electron withdrawing. That affects the energy of the excited state, so how strong a reducing agent the excited state is. Or we could change the identity of this linker, so the anchor. So we could have an anchor where the phosphorus on this phosphonate is directly attached to the aryl on the BIPI ring, or we could put a methylene spacer in between there, and that's going to affect how strongly these couple to the surface. So we're changing 2 parameters, but in a systematic way. When we do that, we see that if we have an electron withdrawing group, so this was the bipyridine one, or the brominated one, that one had the lowest or the least negative reduction potential, so the lowest energy reduction potential, it injected the slowest. When we had an electron donating group, it injected the fastest. And we had this really sort of consistent, so this is, you may not have seen this before, this is log on time because we have multi-exponential kinetics here. Sometimes it's better to show these things so that you can see all the time base and compare them to each other. But one of the things I want to point out is these lines, these fits to the data, these are all simple bi-exponentials. So there are only 2 time components included in these fits to these data. If we extend it to the whole rest of our series, so this slide's about to get more busy, we see a consistent story. So we see faster injection when we have a more electron donating substituents, and we do see faster relative kinetics if we are directly bound, or slower if we have that methylene spacer in between them. So how do we model this to try to understand electron injection? We're going to use Marcus-Gerischer. So the Marcus-Gerischer theory allows us to model the energy of the excited states and their reducing ability, like how strong a reducing agent they are, against the population of conduction band acceptor states. So I like to call this graph math because we're quite literally multiplying graphs together. Now, the functional form for this, I'm going to put it up on the screen, is a bit complicated. But the components of it, we can think about simply as we've got a part of the equation that describes a distribution of injection rate constants as associated with the energy of the excited state as it's spread out relative to the reorganization energy. If we are very careful in the selection of two dyes, for example, then our reorganization energy should be the same. And what we would imagine is if we change the reduction potential for each of these, then we should see a consistent change with the population of injecting states here. So the only thing different here is the relative reduction potential for the two sets of excited states. And then that shifts this distribution of injection rates. When we consider the population of acceptors, so this would be if we had one acceptor at every energy, but our conduction band, those empty orbitals, that isn't exactly a flat or uniform set of states. Instead, what we have is actually a distribution of states. So as we go higher in the conduction band, we have more and more and more acceptor states in the conduction band. And when we multiply this by the conduction band, what we get, so the density of states for the conduction band, what we get is basically a weighted Gaussian, where the weighted Gaussian reflects the population of conduction band states that each of these excited states is sampling. When we integrate those two functions that we get out, what we get are basically just a K observed. And so these are real data, actually. And for an electron donating substituent, where it's a stronger reducing agent in the excited state, it samples higher up in the conduction band, and so has a faster injection rate. At the same time, with our electron withdrawing substituent, we see a slower injection. When we apply that to our whole series of compounds that I showed you, what we see is, well, it actually fits really, really well. So this is our data. These are our, like I said, by exponential. So these are our slower time constants. We plot those slower rate constants as a function of the excited state reduction potential, and then we fit them to the Marcus-Gerischer theory. We can do it for both series, but we do see an offset for the two series. One of the neat things is the slope of this actually matches our empirical density of states. So we measured the density of states in our TiO2, and they both have an identical slope. And the offset between these two is a factor of 4.6, which actually reflects the difference in coupling constant between our two series of dyes. So the one where the methylene spacer, it injects on average roughly a factor of 5 slower, or based on what we would expect from the energy of that excited state. So that, what I just showed you is the time constants for the very last step. So going from when almost all of them have injected to when the last of them have injected. So that last nanosecond of injection. But what about earlier time points? Based on the limitations of our experiment, we could look at these faster time points, and we do have data for them. And so what can we do to make sense of these faster injection rates? So what we thought was, okay, if we excite this thing high up, we know that it injects very quickly. And that was work done by a different group at UNC and others before us. But we see what we would imagine happens is we have this relaxation process within the manifold for our dye. And then we have injection. And this is what we expect is our slower injection. So what's giving rise to this faster injection? And we wonder, is there a population of excited state that there's something going on where it's sampling higher up in our density of states? And so what we see is a faster injection rate because of that. The kinetic model for this would be, well, we have our lower energy state, it injects and we get charge separation, or we have a higher energy state that injects and we get charge separation, but then there's a kinetic process that couples these two things together. If we have this as our kinetic process, does this support our bi-exponential kinetics? Now, digging in, so when you do a fit to any kinetic data, you can define in whatever software you're using, fit the amplitude and the time constant, and you can nudge parameters, and you can make sure that you don't have highly correlated fitting parameters. But ultimately, does it mean anything? Does it have a physical meaning associated with it? A simple bi-exponential looks like this. There's an amplitude and a time constant, and then some sort of, well, that's your time variable, right? So the one exponential and another exponential. If we take this kinetic model and we do some algebra and calculus, we can get an analytical solution that reduces to a set of amplitudes and a set of exponentials and matches the form of our bi-exponential. So contained in this, we have our injection from the higher state and our injection from the lower state, and also this conversion between the higher energy state to the lower energy state. If I use this model and I plot my data, so I plot the fast time component as a function of the fast amplitude, I can fit this looking at the sets of parameters that it confirms for us that these are kinetically coupled. Now, the range of parameters, there are a range here that I'm showing, is between 50 and 100 picoseconds for that, whatever this process is, from a higher energy state to a lower energy state. And that population term is what is essentially here. This population term is somewhere between 0.6 and 0.7. So that's saying from our higher energy state, roughly 0.6 of it is injecting with this competitive injection process, or sorry, this competitive internal conversion process. Or it might be internal conversion. That was a question we've had. If I take my faster time components and I plot those and I apply my model, we can see some of those time constants for whatever process is happening here. Those seem to fit, some of them better than others. So it doesn't seem to be a very fast process. So it doesn't seem to be like a vibrational relaxation or something like this. Our hypothesis is that it's actually the fourth state postulated by Glenn Crosby, and I think Henry Gaffney back in the 80s, 70s or 80s for the ruthenium tris-bipyridine. Our other set, we have a lot more scatter for our methylene spacer ones, and so it's tough to see, but perhaps that one also follows similar time components. Similar kinetics. When we model this, what we can do is pull out essentially a difference in energy between our higher energy state and our lower energy state. And what we get is a energy difference of 0.18 EV, or around 1400 wave numbers. And that is consistent with one vibrational mode of a bipyridine. So it's not entirely clear what's happening, but what's really interesting is this model fits and is predictive. So talking to Jerry Meyer, this would have been January in 2020, shortly before stuff happened. He said that they were able to put on trifluoromethyls onto this thing, so much more strongly oxidizing. And where they saw injection actually fit onto the model. So it was really neat because we now have this predictive model for being able to design dyes and figure out how quickly they would inject. So for this upper end, if in the fast injection regime is something that's important to us, we could modify the structure of the dye to get us there. So ultimately, this model of electron injection comes down to the idea that we can excite our dye. We see a range of injection events, and those range of injection events reflect something that's happening within the manifold, sampling different regions of the density of states. And what's really interesting is the measured time constants for these on the order of a picosecond are consistent with sampling basically an exponential density of states higher in energy. So this entire model is consistent. And there are other interpretations of injection, and people have brought up valid points about the validity of those models. I want to say the reviewers that commented on our paper had a lot of really good criticism. And one of the things they suggested was, well, if something's going on here, then it's intrinsic to the dye, and you should see it if you put it on a surface that you can't inject into. So if we use zirconium oxide, the band gap's much higher and the conduction band is much higher, and we don't see injection. And we did see a 100 picosecond time process on the surface of the zirconium oxide. So it is really interesting that we see this, and it's not entirely clear why it behaves that way or follows these kinetics, but we were really happy to come away with a predictive model for it. Okay, so let's see. I've been going for a little while. I have a few more slides that I'd like to talk about, and really this relates to the work that we're doing at Cal Poly right now. Most of this isn't published. Actually, this is manuscript in preparation, but it's actually been submitted now. So I'm going to share aspects of it and talk about what was recently just recommended for funding from ACSPRF. So I don't know if you noticed, but ruthenium was the theme of the last part of the talk. And as it turns out, ruthenium as a choice for a dye is, well, there are some drawbacks. One, it's really expensive and it's rare. It is the 77th most abundant element out of 91 naturally occurring elements in the crust. First row transition metals are cheaper, more abundant, but generally they're pretty lousy as far as photosensitizers go, and that's in part because they undergo relaxation processes through ligand field states back to the ground state really quickly. That was up until, well, a few years ago, and Lily Sundström and Kenneth Wannemark and their group in Sweden had published about an iron-containing complex that had an excited state on the order of a few picoseconds. Now, to put this in perspective, the lifetime for iron tris-bipyridine, so this is the iron analog of ruthenium. If you look at the periodic table, like, why not just use iron? It's cheaper, right? It's actually a lot less expensive. Well, it turns out the lifetime for this excited state is on the order of 10 to the minus 14 seconds. And not a lot can happen in 10 to the minus 14 seconds. So basically, you just get hot semiconductor, right? Or hot TiO2. So what they found is if they use these carbene ligands, they can change the ligand field states. So to shift those ligand field states to higher energy, and in doing so, extend the lifetime of the excited state here. And more recently, it was the same group published in 2018 in Science, this iron scorpionate complex. So again, using these carbene ligands on the iron, but through basically using symmetry here, they were able to tune this so that the ligand field state was high energy and inaccessible, so they could, at room temperature, show photoluminescence from an iron complex. Now, the photoluminescence here is actually a ligand to metal charge transfer state, which is different than the state for the ruthenium tris-bipyridine. This is also iron 3 instead of iron 2. But the fact that you could have an iron there and this thing doesn't turn dark was actually really exciting. the lifetime that they got was on the order of 2 nanoseconds, which is approaching the time limit for being able to do photochemistry, and is definitely competitive with electron injection. So inspired by this, well, my group was interested in maybe making some first row transition metal complexes where maybe we could see something interesting. I mentioned all of this is done by undergraduates. And so picking something to look at, we have to be sort of careful about the targets that we look for. One of the things, working with Peter Ford at UCSB, I got a chance to work with cobalt complexes. And what's neat about cobalt complexes is they have a higher ligand field than iron. So D6 cobalt has a higher delta octahedral than iron. That means that high spin complexes of cobalt are actually rare because of it. The other thing is cobalt-3 complexes are substitutionally inert. That's one of the reasons ruthenium makes such a good dye is you can make it and then run it down a column to purify it. Well, it turns out cobalt complexes are similarly substitutionally inert, which means in our hands, it's a really useful target. The other thing that we were interested in was the type of complex, and I had gotten into these dithiocarboxylates working with Peter Ford. And so one of the things that I was curious about is if we make these cobalt complexes, can we tune the energy of these states? Can we make things that are more covalent? And so we've been playing around with these for a little while. One of the things that's sort of neat is by changing the substituent group, we can change how it absorbs in the visible region of the spectrum. So these weak transitions, these correspond to the ligand field transitions. And if we have electron donating substituents on there, we see the bands in the visible that correspond to those ligand field transitions. When we put on electron withdrawing substituents groups, we actually do two things. We shift these weak absorptions to the blue, and we shift the strong absorptions to the red. And so these strong absorptions correspond to charge transfer transitions. And we can actually see the colors change based on that. Now, this is a little misleading because this one is actually the weak absorbing one, but my student who was taking the vial pictures made it nice and concentrated so you could see that green there. How to make sense of what's going on here? Well, we've done some TD-DFT and DFT calculations. And one of the things that we see is that the LUMO goes from being basically a D sigma star when we have electron donating substituents to being ligand pi star. And so what we're doing is we're shifting the relative energy of those ligand orbitals relative to the metal orbitals and doing what we would expect should happen in changing the lowest energy state from being a ligand field state to then being one that is metal to ligand charge transfer. So this is one of the things that we're working on right now. Another thing, a paper that working with Taylor Haynes, one of my colleagues, we put together a paper with a whole bunch of undergraduate students. This was a lot of fun to do. Really collaborative project. And I don't know if you've ever gotten to work with an army of undergraduates over the summer, but it's a blast because they feed off of each other. They have a really fun culture. When stuff is working, they are really, really into it. So we're able to make a whole bunch of just the ligand, so the dithiocarboxylates. And we had there were some holes in the literature, and one of the things that we did was model the excited states for those dithiocarboxylates. And in doing so, we were able to characterize that. We modeled it with TD-DFT, and so we were able to make assignments for all sorts of states with these dithiocarboxylates. And just to tell you, they actually have really interesting colors themselves. So they are ranging, well, really from like sort of orangish to red. But what's really neat is sometimes the way they crystallize, you get these neat greens and magentas and other things. Interested in, well, do these have any photochemistry with them? And so this is just one of the dithiocarboxylates. We did some pump probe spectroscopy. This was with Eric Grumstrup at Montana State University. And a little, disappointingly, we excite them and we probe, and what we see is with basically the time it takes for it to dump that energy off as heat into the surrounding solvent. That's what happens. But, this thing doesn't go back to baseline. So is there maybe some photochemistry happening there? And one of the worst things that can happen when you're doing transient absorption spectroscopy is you measure your spectrum before and you measure it after. And you can look at it a couple of ways. You can be disappointed that your sample decomposed, or you can be excited that photochemistry is happening. And so one of my students right now, this is the project that Taylor Haynes and I just had funded, is actually looking at the photochemistry of these things. And so we have this LED array. This is using 365 nanometer light. And so the thing has sort of a red tint. And then after 45 minutes, it's got this blue hue. So it looks like there's something fluorescent in there. And we're going to start digging into what's happening with the chemistry here. Okay, and that wraps it up. That's a little longer than I wanted to go. But I really need to thank the undergraduate students that I've worked with. I think this is all of them. The last time I checked, I think I've had an opportunity to work with 25 or 30 undergraduate students at Cal Poly just in the 5 years that I've been here. At the same time, a lot of the science we do depends on our collaborations. And so I have ongoing collaborations with Peter Ford at UCSB, Malcolm Forbes at Bowling Green, Eric Grumstrup at Montana State, Brian Bellett at Western Illinois University. He makes some really neat organometallic complexes. Josh Furrow-Weiss at UIUC. It has tabletop X-ray spectrometer, which is really neat. John Papanikolaou at UNC. And then Thomas Cheshire and Francis Houle. Thomas is actually modeling our electron injection stuff, but he's using a much more sophisticated model than what we used, and it has had some interesting results, and we had a recent publication with that. And then my collaborators at Cal Poly, Taylor Haynes and Bill Algren. And then finally, my undergraduate collaborators at Cal Poly, Ava and Michael. Also the funding. So first, all of the research I showed you, the first parts of it was funded by the DOE, and that's when I was part of the EFRC. The EFRC is now the CHASE. That's the acronym they have for their center at UNC. And so if you know Jillian Dempsey and Alex Miller, they're part of that center. Jerry Meyer, it's a huge center, and it was a lot of fun to work with those scientists and learn from them. My research with Peter Ford was funded by NSF, and my research at Cal Poly is funded by the College of Science, so startup funds, and the William and Linda Frost Fund. These are alumni of Cal Poly who've been very generous and funded our summer research program, and they've also funded the building that we're going to be moving into. Finally, as of today, yesterday, technically, I got my email that said that our ACS PRF proposal was funded. So woohoo, Taylor and I have a funded project for the next couple of years, and we'll be able to support some undergraduate students over the summer. And those undergraduate students, I got to say, it's been a wild 5 years, and I really appreciate all the effort they've put in, even when they pie me. So at this point, I'm happy to take any questions. Thank you all for listening in.
Well, first, congratulations on your recent successful proposal funding. That's always exciting.
Thank you. Yeah, super exciting.
Yeah, and thank you for a fantastic talk. That was super interesting and really cool. Really cool stuff to learn. Like I say, I was familiar with the ruthenium dyes, but not much beyond the fact that you can stick them on TiO2 and get pretty good conversion efficiencies. So that was really neat to learn about. All right, so we will jump straight into the questions. So the first one here is, does the acidification, the process you talked about at the very sort of near the beginning, when you acidify the dye solution, are you changing the oxide surface charges to enhance the attachment, or are you changing the dye itself?
That's a good question. I don't actually know. I do know that, so it forms an ester on the surface. So it's a titanium oxygen phosphorus ester, if you want to call it that. And it is sensitive to pH. So if the pH of the solution is anything higher than about 1, the dye will desorb. So the equilibrium is definitely shifted toward the adsorbed or the chemisorbed dye under those conditions.
Interesting. Okay. Oh yeah. So you talked about the bi-exponential decay fitting for the transient absorption. In what did the short and long components of the exponential decay actually correspond to?
So yeah, I may have.
You touched on it a little bit, but what I was curious about was is it two separate processes, or is it like a slow injection of charges and then for some reason there's a longer term injection of charges?
So like this model here that we used, this is basically saying, you generate a population of excited states,
mm-hmm.
and then as that population is relaxing, it's undergoing whatever sort of internal conversion processes, intersystem crossing, whatever's happening within the dye,
mm-hmm.
and it's dumping energy as heat off into the bath while it's doing that. When you decrease the energy of the dye, it changes its relative reducing ability.
Okay.
And if you change its relative reducing ability, it's actually sampling fewer states in the conduction band. And so really the difference in injection process is the higher energy state, this is how we're modeling it, the higher energy state is one that is sampling more of the dyes.
Mm-hmm.
It's sampling more of the conduction band, so there's a higher probability of injection, which we just see as a faster injection rate. And then when it's relaxed more, when it's cooled, right, thermally equilibrated, excited state, then it's sampling fewer conduction band states and it takes longer to inject.
Okay. So it's effectively a difference in available population to inject to.
Exactly. Yeah. So let me see, like this picture here, right? So if you take like what we're saying here, what precipitated this is we made that series of dyes. And with the series of dyes, you're basically taking this whole manifold, right, and shifting it up and down in energy.
Right.
But we aren't changing the density of states.
Okay.
So why would the different energy dyes inject differently? Well, it's just a free energy thing. It's,
mm-hmm.
you decrease this and then you're sampling fewer of those states. And so you see a slower injection.
Okay. That makes sense. Let's see, we've got a question here asking, have you tried using vanadium as your center?
I haven't. And that would be fun. It turns out my colleague, Dr. Haynes, really likes vanadium. So we team taught the inorganic chemistry lab, and like he makes these really neat. He makes some carbene ligands, and he also, like, the precursors for those carbene ligands, they make some imines. And he had students in the teaching lab making these weird ligands and attaching them to vanadyl, and then we were attaching other stuff to the vanadyl just for grins, and to have the students measure, like, we were having them measure EPR spectra. So yeah, that would be really neat. I would love to play around with different metals for sure.
Okay. Yeah, the vanadium in particular would be interesting because it has so many different oxidation states that it's at least fairly happy with. So that'd be kind of neat. So I had a question about, so with the cobalt, the cobalt dye that you showed towards the end, you had it with the dithio. The dithiol, yes, that one.
Dithiocarboxylate. It's carboxylate with sulfur.
Okay.
Yeah.
So if you did, like, instead of the sulfur, let's say if you had like an oxygen bonding across there, how would that change? How would that change the properties of the dye itself? So
what makes this kind of interesting is that the sulfur has a really strong nephelocetic effect. So it's weird. What we see in the UV vis is consistent with, basically, instead of having those d electrons localized to the cobalt, they're sort of delocalized off onto these ligands here. But Josh's group, Dr. Vera Weiss at UIUC, like his group, when they did the X-ray spectroscopy, what they saw is this basically reduces to relaxes to the ligand field state. So it's weird because it seems like, with the sulfurs, we would have a more covalent interaction, and your electrons are more delocalized over that sort of like sulfur cobalt core here. And with oxygen, you'd imagine they would be much more localized. I mean, that's what people see when you have like cobalt ACAC, the triacetylacetonate complex is a pretty famous one, and that one does a ligand to metal charge transfer, and the ligand field states do things like the oxygen comes off and comes back on, and so with the oxygen there, it's not as strong a ligand, and the behavior is more like instead of like the dative more like a dative interaction between the metal and the ligand. It's still substitutionally inert, so there's good. Cobalt-3 is a like classical hard acid, and those oxygen containing ligands are classical hard bases, so they're generally pretty strong.
Okay.
And actually, when we make cobalt complexes, one of the challenges is getting the water off of them. So if you want to make like a heteroleptic cobalt complex, this is a fun one. You can make like cobalt bischelate, so like bis-bipi dichloride. And then to get those chlorides off, like the best way to do it is boiling triflic acid. Yeah, like neat triflic acid, like melt your face off, good stuff. Nice. And what happens is it actually protonates the chloride and drives off HCl.
Oh, fun! That's always
real fun. Yeah,
I play around with some of that in my lab, and it's it's something.
Yeah, no, like I have a healthy respect for triflic acid. Yeah,
oh, yeah, absolutely. Yes. So I guess I'll ask one question real quick. I'll insert one question kind of along the lines of the vanadium one. How does the I'm trying to think of the right way to say this. How does the oxidation state of the metal center, or I guess, does the oxidation state of the metal center matter? In terms of what the dye is capable of doing?
Like for the longest time, the way people approached this problem was, that almost like the oxidation state was some sort of canon, right? Like that was important. And this paper, I think got people thinking like, oh wait, I don't need iron 2 to make a functional dye with iron in it.
Okay.
Right. Like this paper, they were looking at iron 2, how do you make something that is analogous to a ruthenium dye but has iron in it? Because iron is I think literally 1,000,000 times less expensive than ruthenium.
That sounds about right, yeah.
It's like $4 a ton for iron, and like ruthenium, I mean, unless you live in Chapel Hill, ruthenium is really expensive and hard to come by.
Yeah.
Well, I'm pretty sure it's still expensive in Chapel Hill. There's just a lot of it.
They just have a bunch of it already. Yeah.
Yeah.
That's right. Okay. And so our last question that we have here, unless somebody wants to drop one in, please feel free to do so. It's more of a comment than a question. It's actually a recommendation. It says look into Tim Hubins. I'm not sure how to pronounce that last name. Hubin. His work from Southwestern Oklahoma State University. Apparently, he also works with undergraduates and also does a lot of inorganic metal complexes. So, maybe an opportunity to collaborate. I'll give you that name again after the talk is over. But thank you. That's actually from one of our viewers, Dr. Rama Kothapalli. She's at Oklahoma, and usually asks really great questions, and also usually has very good advice too.
Awesome. Yeah. Thank you.
So I think if anybody has any questions, get them in. But otherwise, we'll go ahead and start to wrap up. And I'll say again, thank you for a fantastic talk. That was, like I said, this is really cool stuff to learn about, especially the way the design of the die matters. You know what I mean? Like I said, I've had some experience with it, but it's really neat to be able to see all the detailed work that you've been able to do with them to understand better how that interface works and how it affects the ultimate overall performance of the device. And that's just really cool to see. And of course, you're continuing to do great work. That much is clear for especially being able to do it with undergraduates. That's a really cool thing for them as well. So thank you again, David, for giving a talk. We really appreciate it. And I think that's going to be it. So I'll say thank you to everybody that came out to watch. Dave, if I could get you to hang on for just 1 second, we'll talk just very briefly.
Okay.
And to everybody else, have a great weekend. Stay safe. Get a booster if you need to. Otherwise, we'll hopefully see you out here next week when we have, Patrick Julian from the University of Notre Dame. He's a postdoc there, and we'll hopefully have another great talk next week. In the meantime, thanks again, everybody, and we'll see you then. Have a good night.